Epistasis is very interesting topic under genetics. Epistasis is a kind of phenomenon that is generally categorized under post-Mendelian genetics because all the Mendelian rules, Mendelian phenotypic or genotypic ratios cannot be properly implemented in the case of epistasis. It's a kind of intergenic interaction where two or more than two genes interact with each other to produce a separate kind of phenomenon, and this kind of intergenic interaction will be visualized in case of epistasis. To make the concept of epistasis, the basic concept of epistasis clear, we need to go through this simple definition at first. Let's go through the definition of epistasis at first. Epistasis is a phenomena in which an allele of one gene masks or suppress. Put a special emphasis on these words, mask or suppress the phenotypic expression of all allele alternatives of another gene located at a separate locus. What is the inner meaning of this definition? That means one gene which is located at a separate locus, that locus might be located in a separate chromosome, or the locus may be located in the same chromosome but at a different place, right? It is a phenomenon, that means epistasis is a phenomenon in which an allele of one gene, consider the gene is A gene, gene A, an allele of one gene mask or suppress the phenotypic expression of all allele alternatives of another gene. Imagine there is another gene, gene B. A gene can mask or suppress the phenotypic expression of B gene, which is located at a separate locus. Now, how can we understand these things much more easily? Imagine this B set of gene has some allelic alternatives like capital V, small v, that is heterozygous condition, and small b. And all these different genotypic combinations may have their respective phenotypes. But remember, this particular gene, gene A, can suppress or mask all the phenotypic expression of B gene. Whatever different phenotypes these different allelic combination may produce can be masked or suppressed or supervised by this A gene. And after masking the phenotypic activities or phenotypic features of this B gene, this A gene will produce another new set of phenotype that is not present here. It may produce a new phenotype, right? A new phenotype. At that situation, we may say that this A gene is epistatic gene and this B gene is hypostatic gene. That means the epistatic gene is masking the effect of B gene, that is hypostatic gene. Now we will clear our concept with the help of a good suitable example. Okay. We will consider the plant squash and consider its color, squash color, to understand the example of epistasis. Squash is a kind of vegetables that is known as lau in Bengali, you all know that. So squash is such kind of vegetables. Those are cultivated in western countries. Squash generally comes with three color variant. One is white color, another is yellow color, and another is green color variant. Okay. For the determination of squash color, there are two separate genes. Those are responsible, gene A and gene B. Let's write it down. One is gene A and another is gene B. Both these two types of genes interact with each other to produce the color of this vegetable squash. Okay, in case of gene A, there are three allelic variations, as we all know that capital AA, small a, that is heterozygous condition, and small a, homozygous recessive. This dominant allelic combination, both the homozygous dominant and heterozygous dominant, produce the yellow color in normal condition, and this recessive allelic variation produce this allelic variation produce the green color in the vegetable. Now, both these phenotype, both the yellow and green phenotype, can be controlled by another set of gene that is known as gene B. And here, in this example, gene B is the epistatic gene. Okay. Now, gene B has its own combination, own allelic combination, right? Capital B, capital B small b, and small b small. Here, in these two variants, that means in the variant, allelic variant, where the capital B is present, even in a single set, that is heterozygous condition, this both the variant can mask these green or yellow color phenotype. Both the variant, that is homozygous capital B or heterozygous capital B, can mask the phenotype or suppress the color of yellow and green, and can produce another separate color. They produce another completely other type of color that is white in nature. That is, whenever the capital BB or capital B small b, whatever the genetic combination is there, is present in that organism, then even this combination of gene A or this combination of gene A cannot produce their own respective phenotypes. They will always produce another completely new set of phenotype. That means, imagine there are several different type of allelic combination, like capital AA, and we are taking another set of gene of capital BB. This allelic combination, and many others like capital A, small a, capital B, there may be capital A, capital B, small b, there may be capital A, capital B, small b, there may be small a, capital B, and small a, capital B, small b. All these 6 different genotypes. See, carefully see, there are 6 different genotypes. I have written combining both these allelic variations of both these gene A and B. So all these different genotypes produce the white color squash, because here the dominant allele of the gene B is present in homozygous condition or even in heterozygous condition. So all these genotypes get turned into white color. Then when and how green color or yellow color squash can be produced? Yes, they can also be produced in another completely different set of allelic combination, and in such case the recessive set of gene B must be produced. That means capital AA, small b, or capital A, small b, both these two types of genotype can produce yellow color squash, and small a, small b. This type of genotype will produce the green color squash. So here it is the recessive set of the gene B which cannot mask or suppress the genotypic or phenotypic expression of the gene A, but the dominant gene of gene B, that is the dominant allele, can always suppress the phenotype of gene A. This is a good example of dominant epistasis, right? So there are another different example of recessive epistasis are there in my study material. Those who are not availing that particular study material right now, you can go through the example provided in the study material and you can easily clear it out. What is the recessive epistasis, right? So up to this, I think you got clear idea over the concept of epistasis.